Good evening, everyone. It is the 6th of March, 2025, and we are doing a journal club today on a paper, Influenza A virus rapidly adapts particle shape to environmental pressures. Today's speaker, or at least the person who is going to be the guide, we got to come up with a good name for this because I don't like presenter. You're not presenting. It's more of like the person who is driving it or whatever. We should come up with a name. Is Colin Shepard. He is Morris Catholic High School class of 2025 and an AMI exile. All
I'm
right.
still in the AMI, only one out
Oh,
of these four. I actually don't even know if Cameron's here right now, but yeah. So let's get going. I'll start sharing my screen. You got it. All right. So this is influenza A virus rapidly adapts particle shape to environmental pressures. This came out of the Ivanovich lab, which is down in Bethesda, Maryland, from Tihana Ivanovich, who graduated from UCLA in 1999, got a 2008 PhD from Harvard, and a 2015 postdoc in Harvard too. Her lab was out of Bethesda, or not out of Bethesda, but out of Brandeis, the university in Massachusetts for a while before she got contracted by the National Institute of Health, and that's who she's working with now, and that's who she did this research for. So basically, this paper goes over what's you can kind of see as an evolutionary process, at least how I interpret it. It's not necessarily a genetic change that you're going to see, but it's more the shape change that's allowing for more productive viruses in which is specifically the influenza A virus. So quickly, I just want to point out some terms you're going to see. MOI is multiplicity of infection. And there are some cell types you're going to want to look out for: MDCK and CalU3, A549, and CaCO2 are cells that the researchers infected with influenza A, and all the other cells you see on the figures are going to be viral. And then there are two types of influenza A virus cells you want to be, or is cells the right word?
for which part? Because there is for some.
So the influenza A, like particles instead of
Viroid.
cell. Viroid, okay.
Viroid, yeah.
So those viroids are either spherical or they're filamentary. And the filaments are they're producers that are kind of allowing the spherical variants to affect and injure whatever they're infecting. So we'll start with figure 6, and that'll be the next slide. So if any of the Holy Cross guys want to do this, this is kind of just a general overview. This is actually the last figure in the paper, but it'll kind of give you a better introduction into what we're going to be looking at. And if you want to give this a try, it's not actual data, it's more just a walkthrough of the process.
So it's slightly easier. You're desperate to do it. So we decided to put this one first because it's kind of the model, slightly easier.
All
Anybody
right.
feeling it? We should make Miss Scarfone do it. She just got her. Hello.
Hi.
Um, no, go ahead, Colin.
All right. So we'll start in part A, this general model of how influenza A is proceeding throughout whatever it's infecting. So lower output to input and the antibody binding, or it's higher output to input in that beginning stage. You see, can't really point it out, but that first, or yeah, it is part A. So you can see there's more filamentous viroids on that left side and more spherical viroids on that right side. So the left side, lower output to input, so that's antibody binding, and the right side, that's higher output to input because those spherical viroids are spreading more of the virus to what it's infecting. And down to B, so genetic model for pleomorphy and that pleomorphy and adaptation. This is really what you're going to see kind of across all populations that are evolving over time. So first, it's attenuation restricting the population. Variants are selected. There's a little bottleneck there. the filamentous variant here is going to persist. That's essentially just the advantageous trait they want to preserve. Then the escape mutants are going to remain filamentous. There's going to be another genetic bottleneck, and the spherical escape variants are going to be selected, and then another genetic bottleneck at the end. And there you can see in that middle there's that higher output to input ratio where there's the more spherical virus than there are the filamentous ones. Then enough to see there's this, it's essentially the same model. It's the filaments persisting under pressure, the pressure selecting for escape mutants, and then they escape from the pressure where there's that more output to input again, where there's the more spherical virus than the filamentary ones. Filamentary or filamentous?
I'm pretty sure it's filamentous because I think that's what it says in there. I don't
Okay,
know if that's like a bonus actually.
I think it's both.
I saw
Well,
filamentous.
Yeah,
that's
Okay,
kind of
kick it
that's what I started with, but okay.
yeah,
So
I
we'll
think
stick with that.
filamentary is a classification, so I think it works too. I think both of them work.
All right, cool. Then we'll jump next to figure 1. All right, David, you want to try this one?
Yeah, I could try the first half of it if someone wants to jump in, provide some assistance for the second half. But
Right.
so part A of figure 1 is basically showing the filamentous versus the spherical how are we referring to them again?
Doing viroid.
Viroids. All right. So you can see that the larger viroids, the more filamentous ones, scatter that ultraviolet light a lot more. You can see kind of that wider spread versus the spherical ones, which appear to be a lot more narrow. And part B is essentially just comparing the top half, just an image comparing the sizes of them. So obviously the filamentous ones are significantly larger than the sphere shaped ones. And then I wasn't sure what the second half of it was. I think that's also just the spread of the light. On the lower half you have the image, but then the graph to the left, if you move the mouse left a little bit. Yeah, so I think that's also just showing the spread of the light relative to the size of the
Yeah.
relative to the size of them, yeah. But then for part C,
Can I jump in for a moment and just
Yeah.
provide a little background on this? So if you can, some of you may have had this at some point. Like I mentioned, I had to have some, I was in the hospital, last week or the week before, and I had to have some blood work done. And they do the blood work in two different ways: a manual way in which they actually put it on a slide and they count it, or an automated way where they're using a cytocell meter counter, right? So they use a cytometer, and it usually has some sort of easy way of tagging them with some fluorescent marker or something, and then it flies by a camera that goes and counts them really quickly. This is kind of doing that. So that when you flow a liquid full of some cells in front of one of those counters, it's called flow cytometry. And this is kind of a version of that where they're using light, and the sensor is checking whether the thing that is going through is scattering the light wide or narrow. So let's make connections, especially like the younger kids. What color were they using and why? What's it called? This is where it's helpful to actually have your paper where you've actually gone and circled all of the figure mentions of the figures. So you can say, okay, where do they talk about it? It's called VSSC. What does it stand for? What's the V stand for? I wonder if it's a color. Oh, it might be a color. Color that starts with V.
Purple.
I hate you. Yeah, I guess it'd be purple because it's violet. Yes. So, and why? Why violet though? Why use violet?
Why not?
Well, one reason might be that it actually takes a lot more electricity to produce a violet light than some other light, like a red light. So why? Why is the sky blue?
Water, because of the water.
No,
Oh,
not a bad guess though. A lot of people think that. A lot of people think that it's like the reflection of all the water on Earth, or
is that not what it is?
it's not. No. Why is the sky blue?
It's a reflection of the violet light. It's just
Yeah,
how we see
and
it.
blue.
Yeah.
Yeah. So because violet light, indigo, and blue are so short on their wavelength, right? A red wavelength is like this, and a blue wavelength is like that, and the violet one is really, really. So that means that it's going to hit more often particles that of oxygen, water vapor, whatever that's in the atmosphere, and scatter. And so you're only going to, see that. And that's why they're using purple light here, because it will scatter uh much more than some other longer wavelength. So they're using purple light, and it's actually kind of brilliant. And the other point I do want to make, if I may, sorry, David, do you mind? And
No,
and actually, let me ask Colin. Colin,
Yep.
what do you think? What do you think the motivation was for even publishing this?
Well, I know the study. I think the study started in 2023. I believe it was when they got all the funding. I'm trying to think of a current event around that time. Short after COVID, I wonder if they're looking for a more, like kind of how you were talking earlier, maybe a more long-term solution to the vaccine problem, and how we're kind of engineering our vaccines to just be temporary solutions and not the permanent solutions that the COVID vaccine kind of was.
Right.
Yeah, I was gonna say, like, to piggyback off Colin a little bit, I think the issue with these virons that are basically like shape shifting is that that means that some of our like antiviral drugs might actually be, encouraging their persistence a little bit, because we're using some of these drugs that are just making them the spherical ones turn filamentous, which means they still stick around, and it means they're still persistent. So I think the study is explaining why that's the case and how we're not eradicating it by threatening it. I don't know if that makes any sense, but
Yeah,
like,
no. I
yeah.
think both of you are right in that it accomplishes that. I don't think that was the goal though. And that's where I want to make I want to make a point, especially for the noobs here. Um, the freshmen are just about to start doing data analysis on their diffusion and osmosis lab, which the Morris Catholic kids did too. Okay.
Yeah, have fun.
Yeah, fun. So that could be a fairly rough thing. It's, I keep hearing it's the hardest assignment they have, period, in all of high school, in all of freshman year. But one of the weird things about writing a manuscript is the order in which you write it, right? And so with the freshmen, I haven't gotten this yet, but certainly the sophomores had it last year, right? You write it completely out of order that you read it. So you write it. What's the first thing you guys do? People who know.
Results, I believe.
Yeah. Well, figures and
Yeah,
tables,
figures. Yeah,
which are results, right? So
yeah.
yeah, and you write the results, then you write the discussion, then you write the intro, right? Somewhere along the lines, you'll write the materials and methods. So when you end up writing the hypothesis, you write it as if you knew all along what the answer was going to be, right? And in truth, when we're doing these experiments, we often find out we're totally wrong. Like I wrote a grant in grad school. I got $250,000 to do this project, and I had this idea, hypothesis about how the circadian clock worked. It was completely wrong. Now, they're not going to take my funding away just because I'm wrong. It's just that it worked a different way, right? But when you read the paper, you wouldn't know that because we hypothesized that, and as if we always knew it was that. So there's a little hint. There's a little hint in the paper, right here. We developed a quantitative flow virometry assay. I'm having trouble. We developed a quantitative flow virometry assay to measure the shape of viral particles under various infection conditions while using different combinations of blah, blah, blah. Did anyone notice that?
I did. I took a note on where it said the flow virometry because I wasn't totally sure what that was, but that analyzes the single particles.
Yeah. They're going to make some money off this. I think that's the actual driving force behind this was they developed a new method. And it might also be the reason why it's not in Nature. I'm not sure. I'm not a virologist. But they developed a new method, and kind of one of the annoying things is like you all know that picture of me. I'm telling mostly stories from my experience, obviously talk about what you know. You know the serial killer photo of me with like the red light and stuff? And the thing I'm holding was something I invented. And it was the first time that which one was it? The Journal of Neuroscience paper I published in 2008 or 9, I think it was 9. Yeah, it was 9. Was the first time that was used. And that was kind of the point. We had developed a technology that we wanted other people to use and, shortcuts for how to do things. But methods papers don't really get you anything. So you kind of actually also have to study something. So we talked about vasoactive intestinal polypeptide. Um, and Amber, who's a senior at Holy Cross, she's studying something related in Keflin. And so it's not always this pure we're all saints and we're all doing thing for the good of science. No, it's possible these guys are making money off of this. Once it's probably going through a patent right now, and they just needed a study of something as a proof. It's called proof of principle, right? Just to prove that it does work. I had another study where my sister and I came up with a way to monitor mouse behavior using a security sensor. And we had to decode like the different breaks and how that would work. That's not a publication. That's not interesting. But if you use it in an actual study, now you have other people started asking me about it. Hey, what is this? How did you do that? Can you set it up in my lab? Make sense? Is that interesting? Do you
Yes.
people like getting that sort of background insight? Because I'll shut up if you don't want it. Huh?
I like
Okay.
the background.
For it.
So
I
I
enjoyed
also
it.
who was that?
Very cool.
That was Cameron.
Oh, can't. Okay. Oh, you're here.
Yeah.
Tyler. Hello, Tyler.
Hello.
What's up? Somebody down there is Zoom user. Whoever you are, I need you to put your name. And so, unless there is some sort of catastrophe happening or something that's really distracting in the background, we would like you to put your cameras on. So, Cadence, you should have your camera on. Morris Catholic kids, you don't have to do that. I have no
I
power
put
over
mine
you.
on, but my camera won't work right now.
That's okay. So if you have a reason, but Zoom user is worrying me, you might be a terrorist. Who are you? Okay. Whoever Zoom user is, I'm kicking you out. You can come back in as your actual person. Remove. Remove. When COVID happened, we had, we went online and we didn't really have a protocol for staying secure. So like I'm not using it right now because I can't manage everything. But the I remember in the first week of the COVID thing, we were doing Zoom, and I guess one of the classes for one of my colleagues at Morris Catholic was not behaving very well. So the president of the school went into the room, into the Zoom chat to like kind of give them a scolding. And then somebody that was not a Morris Catholic kid came in, yelled at him with profanity, and then just like left. And
Yeah, I saw that video. Someone
yeah,
was recording at the
somebody
time.
recorded it.
Yeah.
That makes its rounds at least once a year.
Did they make fun of him for like being bald or something? I might be
Yes.
here.
Yes. And unlike with me, where I actually joke about that all the time, he was, that kind of upset him. So
Yeah, you
that's
might
not
be
very
the
nice.
next victim.
Well, I just kicked out whoever Zoom person was. All right. Okay, so I'll probably cut that out of the video, but and the videos, guys, are actually already up on the website. Okay, so back to David. I interrupted you for a long time. I'll go back to sharing the thingamajigger.
Yeah, no problem. That actually was a perfect little stall. Gave me more time to stare at the figures and let them click. So, all right. So, looking at parts D through G, it's basically taking a look at two different strains of the IAV virus. So you have the PR8 and the XUDON, and they injected them into two different cells, which was the MDCK and
What are those things?
CalU3. Which
What are they? I know one of the MDCK is something kidney.
No, what are they? Are they cells? I don't like. I
Yes,
couldn't
they're
figure out
the,
what
Yeah,
that was.
yeah,
Either.
those are live. The PR8 and the XU-DORNs, those are
Strains.
influenza A strands, yeah. And the MDCK and the CalU3, those are live cells that they're infecting.
Yeah. And so basically what they did was they injected them into the into those two cell types. And looking at the graphs, the leftmost ones or I guess we'll start with D. This is basically taking a look at the yield of how many, I guess, virions were produced. That's the same thing for the first graph in E, F, and G. And you can see that relatively similar amounts were produced within the same time frame, regardless of the strand. One important point of clarification also, the PR8 strand, I believe was, I think, mainly spherical, whereas the PR8 strand was mostly spherical and the XUDON was mostly filament, or filamentous, or however they're saying it. So the overall amount was pretty similar, but when you look at the shape and the percentage of filament, for D, we can see a sharp drop in the percent filament immediately following the infection. So from like that, I don't know, 5-hour to 20-hour range, you see that sharp drop, implying that most of it was spherical. But then shortly after, you see it start to grow and most of the strands become filament. And yeah, that happened for D. Similar in E, although it was mostly filament throughout the whole thing, just because the XUDORN was mostly filament to begin with. And yeah, you can see for figure or parts F and G, it's mostly spherical throughout the entire test period, I guess, or whatever's listed on the data. Yeah, that's all I had. If anyone wants to clarify anything.
All right, yeah, no, that's great. So yeah, so you can see as the trends that they have pointed out, that as there are more spherical viroids, with the exception of E, you're seeing far more or far much more yield on those left graphs compared to the right ones, where that filament percent is decreasing again, except in that XUDORN where they already have that higher filament percent, as David pointed out. All right, we're good to jump to figure 2. Lila, you wanna jump in here?
Okay, so just for a little overview before we actually start with the actual figures, this is kind of summarizing stuff people said before, but just in case you wanted like a little bonus, like redo of the context a little bit. So I guess what we know up until this point is that something's influencing like the viron's shape, either like spherical or filamentous. Spherical we know is like good for division. and filamentous is good for like, being more defensive. But and we know it's now
What do you mean
like,
by that?
What do you mean?
You said good for being defensive. What do you mean?
Okay, well, I guess what I mean is that
I'm not defensive. Why are you being
No,
defensive? No. What is?
no, I think filamentous is I don't know why it's less likely to be eradicated. I think filamentous it's like less individualized because I think filamentous is like multiple particles connected or something like that. I feel like the shape was easier to pass through and stuff like that. I don't
guess
I don't remember.
I think that's a reasonable
harder to break
interpretation.
down big versus small.
Yeah, that because I think it's either
No,
a bunch
so-
of
Colin, what do you mean by that? It's harder to break down by what? What does that mean?
That was David. That was zero.
Oh, that was well. I'm saying like antibodies.
yeah.
I'm just assuming like a larger viroid, it would be tougher for the antibodies to break it down and kind of damage it versus the smaller ones, which can rapidly kind of multiply.
Looking back at figure 1A again, back to the violet side scatter. What I think was helpful for me to understand that was I was kind of looking at it as a measure of chaos, where kind of like an entropy type thing. I don't know if you Holy Cross guys have gotten to that yet. I think that's more like a chemistry concept of like trending towards chaos. I remember Dr. Char mentioned that a lot in our
Yeah, what law is that, freshman? You people who are afraid to speak, you can hold up your fingers since you're not willing to speak. Which law?
The second law of thermodynamics.
That's right. That's right. Like sophomores too. I teach this class completely different than you guys. The whole first quarter, we did no biology. Yeah. Yes, Devin. Yes. It was because of your class. I was like, I have to reset and teach a different way. And so we were trying to do discussions and try to get them interested in asking the right question and stuff. So that comes up a lot in those conversations is like the entropy and the thermodynamics. So
All right, Nalana, if you want to proceed with figure 2 now.
Totally took the wind out of my sails. Thanks, Dr. Char. Okay. Yeah, so basically figure 2 is all about trying to figure out what it is reacting to like change shape. So for figure A, I think that's just supposed to be so I think it's figure A and B are figure 2A and 2B are similar. Because I think 2A is just the x-axis is the
Yeah, they do just flip the axes, yes.
Yeah, they just flip the axes. So for A, which is the one I'm basically going to be focusing on, the cells are at the bottom. And I think the point of this one is just to show that it's some cells have more of one type than the other, but it's not totally connected to a certain cell type. So like MDCK has more, I think the most like HK68, which is a filament, and then CALU3 has more Cal like 0709 by like a smidge, and that one is more spherical, but it's only by a little bit. And then for like A549 and for CACO2, it's more mixed. So that's kind of just showing that the cell type doesn't matter as much. And then I think for C,
can I pause you again?
yes, oh sure.
So again, sophomores. Are there any juniors here? I didn't see any on the list. When so with the freshmen today, I started statistics. So tomorrow, those of you who are in, was it F block? I'll have you do statistics again. And we were talking about lies, damned lies, and statistics. And in this case, Lila just said that it might look like something, right? You said there was like something you might notice, or it sort of looks like I think that might have been how you phrased it. We'll have a video evidence of it, but it sort of looked like whatever. And I want you to be really skeptical about that, because if it's not significantly different statistically, then the things you're seeing as differences aren't real. Right? Right?
Yes.
Because that's the whole point. Like I could say, all right, you seem to make an observation. is the observation real? And so, Lila, is your observation actually real? Is that true?
You're saying my observation that the cell size or like cell type doesn't matter that much?
No,
Is
no. So what's funny is, and again, we'll have video evidence of this, so I might be wrong. I'll preemptively apologize if the video evidence shows that I'm being a jerk here. But you actually said the opposite of that first, and then came around and said that's that part you just said, which I think that's what they said. And if I read it, while cell line and viral strain biased the shape of the produced virions, no single factor was decisive, and infections produced a range of shapes. So that's kind of what you were just saying, right? It doesn't really matter that much, which would suggest there's no significant difference, right? But earlier you had said that it looks like one's bigger than the other, which in the analogy I gave the freshmen today, and I will tomorrow, is when not running a statistical test, middle school students and high school students will just point at a graph like that and just go, it's bigger because it's bigger, right? You're not really sure though. And if I'm being really hard on the kid I would say probably don't do this. Like if you go to a science fair and you're like, did you run an ANOVA or a t-test? Like that's you just being a jerk. But I can make self-deprecating jokes all I want. And in this case, when we're doing this stuff, I think, did they run a test? Obviously, right? Yeah, of course. It's published in Nature. In fact, I am sure that AI actually tested this again just to make sure they did it right. Right? But this was not a significant result. It showed there wasn't any significant differences. Right? I thought we were going to skip 2.
I thought we were skipping 5.
I thought we wanted to talk about like how, especially in E, how the error bars are way too big. I thought we wanted to talk on the statistics.
Oh, okay. Well, all right. So let me frame that then, because I was responsible for that. So for the freshmen, if you read this, and I would hope you did, so because you're not getting anything out of this at all, just from the banter, you won't get anything unless you actually tried to figure this out. But when Colin and I were talking right before we started, we were talking about which ones should we skip, because you don't need to do all the figures, just the ones that really make the point. And I had suggested maybe 2 should be skipped, except for G, because G's the only piece of analysis that actually was significant. Pretty much.
Who was going to tell me
Right.
that? Who was going to tell me that? Colin, were you going to tell me that? Who was going to tell me that?
This was I think 5 minutes before you started, and you
Yeah,
said
yeah,
you said you were going to.
And then you said you wanted to.
You weren't there.
Yeah,
Huh?
you said that you poured your blood, sweat, and tears into it. So we didn't.
No, that was for
No, no, no,
5.
no.
For 5, I joked that I poured everything into it when I spent like 30 seconds on it, because I was like, who's doing 5? And there were crickets. And I was like, okay, I'm doing 5. And then I looked at it and I was like, okay, so that's fine. That's
Well,
great. That's okay.
When Colin and I were talking about it and trying to figure out if we should just like take it out for the most part, except G, you do sort of, in order to understand G, you sort of need the context of the other ones, especially E. So you're back on the hook, I guess.
I didn't like E. I liked how all the other ones were basically just saying that it's either a weak correlation or no correlation, which is like easy
Right.
because you just like shrug and you're like, okay, anyways.
Well, how do you so if you're looking at this, it's my computer's behaving like a Mac. Right, can you see my mouse? Yeah.
Yep.
So for those of you not familiar with statistics, one type of stat you can run is a regression analysis, which we call correlation, right? Something happens and something else always happens with it. It's a correlation. Or the reverse. If something happens, then another thing never happens. That's an anti-correlation. But the rules of statistics still apply, and this p-value has to be, do you guys remember the rule?
We just learned about the p-value in AP stats.
Best class.
Yeah,
Sophomores.
doesn't have to be less than, what is it, 0.1 or
No.
0.01?
Well, it depends. If you're talking about in mathematics,
in
I
math.
think
That's what we were
they
doing.
do point.
Is this the one
Yeah.
where it had something to do with the number 5? Or am I thinking of something else, I
Yes.
think. Okay,
No,
great.
you're
Because
not.
I
That's
remember
yeah.
No, you asked me a question at Hopkins and I got it wrong, and you stared at me for like 3 seconds and didn't say anything and moved on. And I still like
Yeah.
lay in bed at night and like think about it, and I still don't know what it is. So I'm sorry, I'm gonna mute it.
Yeah, well, it's so I always teach, I start you guys at freshman year so you can appreciate this. So this thing is the probability. P stands for the probability that something is due to chance and instead of being real. Okay. So in order to be considered significant, P, that value has to be less than 0.05. Unless you're in some other field like math, where I think they're a little more lenient and they'll say 0.1's fine. But in my field, it's 0.05. So for C, is that a significant regression? Anybody can chime in.
You're saying it had to be less than.
Yes.
Yeah.
Yes, because it's less than 0.05. So yes,
Yeah.
0.04 is less than 0.05. So yes, but what about the next one? D.
No, that's
No.
way, yeah.
Nope. And then you look at F, no. But then you look at G, and G's ridiculous. But all right, so go ahead, Lila. Now you can pick up.
I think was Colin saying he wanted to talk about how large the error bars were for E, or was that? Okay,
I can
Colin.
just that's like 2 sentences real quick, but yeah.
Yeah.
If you guys notice how the error bars in E, if you look at that one cal bar under the cal U3, it's I guess the 8th one from the left, the middle bar that's sticking straight up. You see that stretches from the highest percentage of change of percent filaments to the highest positive percentage all the way below 0. And you see some smaller ranges in there too, but if you look at even that second one from left, that's halfway into the positive percent and halfway into the negative percent. That's a significant spread that's denoting that it's not necessarily a poor test, but a poor test to include. And that's kind of what Dr. Char and I were talking about before we hopped on.
Especially if you're trying to sell units of a new counting technology, because for only $25,000, I will sell you this new, like I had a grant at Vanderbilt to get one of these, but not for viruses. And I got the grant was like $10,000 and the unit was 9,900 and whatever. So I like just had enough money to buy it. I don't think I used it a single time when I was there. I just got the grant, got the unit, and gave it to somebody else. But okay, keep going.
I don't know if Colin was still talking about his thing. I can talk about it.
All good. Yeah, dude.
Okay. I was gonna say for G, look at that p-value. Whoa, that's so small. That's yeah, times 10 to the -5. So that's like 80 bitty. That's crazy. So I think what that one is trying to say is that unlike the rest of them, where it's basically just meant like a whole lot of nothing, this one is saying I think there's a strong, and I might be getting this wrong, like negative correlation between like infection efficiency and filaments. It's basically like the less successful the virus is, the more filaments it produces. So that's kind of just, I feel like the entire paper in summary a little bit is just saying like the more inefficient a virus is, the more likely they are to be filamentous to like adapt to that. So it's not in response to like cell size or to like infectivity or yield or anything. It's in response to like the efficiency of it at like infecting the cells, I think. I don't know if that makes sense.
Yeah, no, I mean, yeah, negative correlation. Yeah, perfect. I'm interested in what was just drawn though.
Wait, sorry?
I think you're
Sorry, I
muted.
forgot
Yeah.
I'm muted. I am stupid. Yeah, I'm talking and talking. If you draw a line, right, and all those dots are really close to that line, that is a strong regression.
Oh, yeah.
And very, I mean, it's so the R value is almost one, almost
Yeah.
-1, like it's a 1-to-1 relationship. That's pretty impressive. But I'm gonna add another piece of teaching here. We use in biology, we use a cutoff value of P is less than 0.05 to say that something's real. Okay, or significantly different in a real way. If the P value is crazy low like this, like 2.11 times 10 to the 5th. So math people, that's 0.0000211. Is that right?
Yep.
Something like that. Yeah. I'm not good at math. That more significant than this one? I'm asking you a question that your middle school teachers would be proud of.
Yes.
I feel like if you're asking me, you want me to say no, but like,
Correct. That's
okay.
correct. So the answer is actually no, because we have kind of, as a field, arbitrarily said if it's less than 0.05, it is significant. But what if it's a really, really small number? Well, it's still significant. Like, I love when people ask, like, so how pregnant are you? That's a binary thing. Either you are pregnant or you are not. There's no more pregnant or less pregnant, right? Like it's binary. So in this case, you are significant or you're not significant. So then what's the value of that number? The lower that number is, the more confidence you can have that it's real. So the lower that number is, the smaller it is, the more confidence you have that it's a real thing. Okay? Still significant, but you're way more sure of that than the other numbers. Okay? And all your freshmen, you're going to be writing a paper, you're going to be using p-values over the next month or whatever. So this is important for you. Okay. Sorry, I keep interrupting. Go ahead, Lila. You can finish.
I think my queen Maggie is raising her hand.
Yeah.
Yeah, she could go.
Sorry, so I just wanted to ask, what does the R and P value like stand for?
So the R is the correlation coefficient. I don't know if you've had algebra 1 necessarily yet, but that entry level statistics, you're looking for a correlation that's closest to 1 or closest to -1. That shows you how linear the data is going to be, how close it is to forming a line. You see that orange line Dr. Chart drew on figure G that's going straight down. You see that R value is -0.882. That's really good. That's really well correlated in a negative way. For a scientific test in math, you're going to see correlations closer to 1. Like a lot of times you'll get 0.99 or 0.98 or just a standard math problem. But in a real experiment, -0.882, I mean, that's wonderful to look at. And then P is just that significance value where you want to be below a significant value or below a value, below the P value, for the data to be considered real, as Dr. Char was just saying, real or just due to chance. So the lower that P value is, the more likely it is to be a real occurrence and not just some bad sample.
Okay, thank you.
Yep. All right, Lila. Anything
I
else here, or?
I think I was basically done with my figure. G is the important one, and the rest of them are boring. So that's my.
All right, awesome. So we can jump to figure 3 then. Any underclass want to do that? Cameron, do you want to jump in there? Did you get to look at it or no? Here right now.
Yeah, I'm here.
Cam's thrown under the bus thoroughly on that one. He's like, oh.
Yeah, so
So this is a college student right here.
Well, also
was-
he should have joined our
I couldn't.
presentation call, and
I was doing something.
I could, yeah.
I don't know what you think is more important than this, but all right.
I was even trying to read this during that class anyways, so.
If not, don't worry about it. I can just take it. I can have one of the other guys do it, but.
All right.
Okay, anyone interested? Anyone up for it?
Wait, I have a question. Is
Yeah,
that
what's up?
thingy like where it says like transcription or whatever above it? Is that the Golgi apparatus or whatever? Like, what is that?
I do not know. I do not know what that apparatus is. I would imagine that's something that the transcriptions that's getting sent out of. I mean, that would make sense, or it's getting sent out of the nucleus for more coding. I'm not sure.
That's what it is.
Nucleus.
It's the nucleus, yeah.
Okay.
So that's Cadence, your question's not stupid. It does look like that because it's this stuff is an endomembrane system. This is ER, right? That then leads to Golgi. So Transcription happens in the nucleus. We haven't gotten to that yet in honors bio, but yeah, because that's what's weird about like when I said we spent the first quarter not doing bio except to talk about like philosophical, ethical things. One of them was what's alive? What are the principles of life? And the problem you run into is viruses. Are viruses alive? One of those principles was that they're able to reproduce. Do they? No, because look, they actually steal your machinery in your cells to trick the cell into making more of it like a factory.
Yeah.
Maria, have you guys gotten to that in Miss Akers' class? No, right? You're doing cell respiration right now? Yeah. Okay. All right.
Last call, figure 3, or I'll jump in on it.
I can try.
All
I
right.
was just giving it a look. So the first thing I see that it says influenza A life cycle. So like the life cycle of a virus, even though it's not alive. And so we see that it attaches to, I'm assuming, some part of the cell in this endocytosis process. Then you go down and it says fusion. And then it looks like it's kind of injecting parts of it into the cell, which leads to the transcription process where you can see that little strand coming off and transcription that has to do with creating RNA. So it looks like it's replicating its RNA within the cell and creating more of the virus, and then secretion. And I guess budding means that it's releasing more of the viruses out of the cell. Maybe I'm right, maybe I'm wrong. That was my first interpretation.
No, it looks good. I think I'm wondering if below that large, I guess, purple line, if that's the border between the infected cell and the viroid, or if that entire complex is the infected cell and the viroid is that area beneath that kind of nucleus area we were talking about with the transcription before. But I'm not sure.
I'm not quite sure. I understand your questions.
So inside, I'm trying to find if there's a border or where the defined border between the viroid and the infected cell is.
I think the
it's right.
viroid is
Oops.
the circle.
Yes.
And
That's
then
it.
the hagglutinin, that's that the fluorescent thing which is attached to the virus.
That's the British spelling of hemagglutinin.
Okay, that's not much easier.
They added a lot of A's where we don't usually use them in an extra G, but hemagglutinin, it's a protein, and it's stuck to it's one of the things that identifies cells as being self stuff. But a lot of these viruses, they'll either just inject their RNA, and that RNA will then be transcribed, or they will have to actually be enveloped through endocytosis first so that they can go down to the source. Remember in early COVID, maybe you don't remember this because you guys would have been too young, but in early, Miss Garfone might remember this. Remember in early COVID, we were noticing that people with O-type blood tended to have way better outcomes than A and B. And there was a hypothesis about why that ended up kind of being true, which was O-type blood didn't have any carbohydrates or proteins sticking off of it that the virus used to dock. Because O is not actually a thing. It's the absence of. O means you don't have it at all. You're not A and you're not B. So the virus couldn't land as efficiently on the blood cells. Yeah. Anyway, go ahead, Cam.
I don't think I had anything else to say. I kind of had a question though. In the paper, it says, like, I'm just gonna read exactly what it says. The first reference to the hemagglutinin, or however you said it. They said we used a fluorescent hemagglutinin antibody that separates virins from noise. And so the way that I initially read that was them saying that they attached something to the viruses that made
That,
able to view the viruses, or is it
yes.
already on there?
No, that's what they did. What
They
you said is
attached
what they
it.
did. Yes.
Yeah, that's
And
that
the thing
acid
that,
for
yeah,
Science
it's
magic.
a
the fluorometry.
right.
Yeah.
So I can't see it. So what I'm going to do is I'm going to attach a fluorescent tag to it and shine this fluorescent light on it that will help kind of do the sorting faster through a different machine. Then I have a more concentrated solution that I can run the virus through my new machine. At least that's how I interpreted it.
But that leads me to another question though. When
Yeah.
it like reproduces or it transcribes its RNA and produces more of the viruses, if that's not part of the genetic code of the virus because it was added
Yeah.
by the people to just view it,
Yeah.
why does it look like they're producing more of the those antibodies, those proteins or whatever, when it's transcribed and then they're secreting the new ones? Maybe I'm just like, maybe that question doesn't even make sense.
No, I don't know. That's a good question.
Do you get what I'm
Hemagglutinin is-
saying?
Yeah, I do. Yeah, I do. But hemagglutinin is a naturally occurring thing on eukaryotic cells anyway. But the ones they're using for sorting are fluorescently applied with, I believe, antibodies. So
Yeah.
you'll still have. I could be totally wrong about this. So somebody viewing this video 50 years from now be like, look at this moron trying to explain this completely wrong. But it's going to have hemagglutinin anyway. I don't think that it's going to have the fluorescent tag.
Okay.
Okay.
All right. That makes sense.
Yeah. If I'm not mistaken, HA is also the thing. Like if you need an organ transplant, let's call it a liver or something. If you need an organ transplant, and let's call it a liver, then HLA is a similar protein that is required. You need to match between people, or your body will reject it. Okay.
So you're gonna make us all test for if we have the same one as you, so you can harvest one of our livers.
I didn't say that.
Well, when I get the HLA test in the mail, I'll know who it's from.
Oh, yeah. But nowadays you just need a piece. I just need a piece, a small little piece from one of you. I'll collect it myself in a bathtub in some hotel, just leave you all iced up like they do in the shows and the movies.
Well, that's what you do with the students who get the lowest grade in your honors bio class. That's what happened to like
Harvest.
Sarah or what?
All right. So what about the rest of that? Did we do BC?
Cam, you want to look
Yeah.
at that or you want me to jump on it?
Because I wanna kind of pick up the pace or close up if we can.
Yeah. We can probably, we might be able to skip figure 4. I don't know what your thoughts are on that, but
Yeah, we can skip
BC,
forward.
BC, D, and F are just different things they're inhibiting. It's the attachment fusion
Right.
or transcription, and then those two different transcription ones, they're both looking at yield, but the one is plotted with a multiplicity of infection of 10 and the other with 0.3, and they're both using that Bullock's over, and you can see that's a different scale also on that bottom one.
Mm.
All right, we'll probably get to jump to 4. I don't know if we need to touch on E and G all that much. If you even want to jump to 4, that is. So
Yeah,
yeah,
jump
this is
for.
jump to 4.
Mhm.
Okay. All right. Any more volunteers for that one?
For 5 or for
For 4, we were
4?
skipping 5, and that was
Yeah,
just
we'll
gonna
skip
be it.
5, right?
Yeah, we're skipping 5.
I mean, ultimately, this is just showing us that, you know what I thought they did a really good job was, if you're looking at the paper, the conclusions they're coming up with are actually the subheaders. Did you see that? It's like attenuation
no.
after attachment drives filament assembly, and then it's a whole thing where like guys, freshmen, when you're writing your paper, this is a really good model for how to write a manuscript because in the results section, they do exactly what I would instruct you to do, which is you say what the problem is, you use an if-then statement to frame it, and then you kind of end each paragraph with what the result was. Like, what's a good example of that? They did it a bunch of times. Like these results would suggest that blah blah blah. It's really well written.
What's a manuscript?
A manuscript, the word manuscript is just any written document before it's published. So if I have a manuscript for a book, even if it's not in science, right? It's a manuscript. And then after it's published, it's a novel, it's a book,
So it's just like a rough draft.
not a rough draft. It just means it hasn't been published yet. You can have a final
Okay.
draft. Yeah. Okay. Colin, why don't you just
Yeah, I got it. All right. So these are just different treatments. I believe it was with treatment with different antibodies and different for each different cell type. So you can see they're running 2 PR8 tests again in the MDCK and then the CalU3 on that second row, then 1 exudorant test with the CalU3. And then the biggest that takeaway that I kind of had is even with the treatment with antibodies, it's that same continued yield. Like other than you can see in that hemagglutinin head portion, it's a lot of the same yield, especially you see that second column where the hemagglutinin base is and the fusion is. The data doesn't spread all that much. I mean, you can kind of see it a little bit in the PR8 in the MDCK, but in B, all that data is, I mean, I guess relatively close together. It's not great, but in C, in the exudorant, that's really tight in the second column. And then in the third column, that M2, that's really close together, even as treatments are changing. You can see those blue pre-treatments and the red post-treatments, they're maintaining that same yield even after.
Okay.
And yeah, that's pretty much a wrap. And yeah,
Okay.
Conclusions, are they not there? They should be there.
Yep.
All right. So real quickly, kind of what we were talking about earlier, that typical vaccine resistivity is bound to increase over time. So that's kind of like looking for a more permanent solution. This is almost kind of what this draws us to, if we're able to realize how viruses are changing their shape quickly and adapting to their environments without a real significant genetic change. Next, it's opening the door to potential new vaccine types and a more permanent solution, kind of what I was just getting at a couple of sentences ago, but yeah, just paving the way for a quicker response to different influenza A strands, because they do new flu strands come out all the time every year. And yeah, kind of just streamlining the process like we did with the COVID vaccine, which I believe is the third conclusion. Yeah, streamline processes of vaccine development. And that is all. And you'll see that picture from our AMI trip to the Met in June of 2022.
The AMI exiles.
Yeah.
I'm not in that picture because I wasn't
I've never
with the cool kids. I was just there.
I've never seen this picture before.
I forgot who took it, but
who's this
it
person?
was just at our. Who is your face is in the way?
Is that I think that's Cassandra.
Yeah, I think that's Cassie.
Yeah, that's
I just
Cassie.
said Cassandra. Ew.
She's on
Cassandra. Okay, so I saw this, my wife and I just watched this documentary because that's what we do for fun. On the next big pandemic they're predicting is this bird flu thing, right? The H5N either 1 or 2 or whatever.
I think it's 1, yeah.
I think this is going to be really important for trying to identify if somebody has that virus, right? If somebody gets infected with that and you need a fast way of assaying what they have, you could probably do it with this. And that then speeds up, as you said, vaccine development for that. But why haven't they come up with a cold vaccine yet? Right?
Yeah.
Yeah. Did you think, so I try to end with this, well, first of all, do you have any new word that you learned that was
Oh, hang on. Let
word
me.
of the
I bet I can find one within like the first page. I'll be right back. Hang on. I'm
and
gonna look.
then also this nature microbiology, nature, science, cell, eLife, those are the sexy 4 journals. Nature isn't there's nature, but then nature also has a bunch of other child journals, I guess journals that are more specific to a field. Like the one I showed you, my paper was in Nature Neuroscience. This one, Nature Microbiology, has an impact factor of 20, 21 or something like that. That is huge. The paper we covered last week had an impact factor, I think, of 4. This has an impact factor of 20. So if you're in the field of microbiology, you read every one of these papers that comes out in this journal because they're so important. Did this paper deserve to be in it, Colin?
Yes, I think so. And especially how we were talking earlier that it probably could be in the real nature. But again, it's a new process, so it needs to be that. What was the phrase you used? There's product testing essentially.
Oh yeah, product development. I mean, they're coming up with a
product
new
development,
new way to do that, right? Let me see if I can do this without. Hold on. I'm gonna go. Shoot, it's behind.
I'll go
All right,
best new
before you
word
guys
is
go. Yep. Go ahead.
best new word is the British spelling of hemagglutination.
Oh God. Yeah, that's dreadful. Let's see. I want to go to.
I liked
sensitivity.
nation was a cool word. I thought that was cool. That's me. That was in I think figure 6, which we did kind of. Yeah.
What was it, Lana? What word?
Attenuation. I think it just means like when a virus is like weakened due to like mutations or drugs or immune responses or
Okay.
whatnot. Yeah.
Oh yeah, I thought the same thing, Lila. I had to look that word up.
I had to look up a lot of
It's
words.
kind of embarrassing
So
for you, David.
no, it's,
But,
so Lila, you're gonna do next week?
yeah.
Okay.
You didn't like my the piece that was old. You didn't like it. Made me sad. So now I gotta go find one. But I'll
I
find it
didn't
like
like.
fast. Hmm?
I don't know what you're referring to. You didn't like what?
No, you didn't like. I sent you a paper and I was like, it's too old, but like you should like look at it and make sure that like it is too
Oh,
old.
this
And
isn't it? I thought this is the one you wanted.
no, wait, what? No, you mean the one that we just covered?
No, the one on the screen that I have on the screen right now.
Oh yeah, sorry. No, I was on the thing where it had all the faces because I thought we were like doing
Oh, okay.
presentation. I was locked
Yeah.
out. Now I'm
So,
locked in. Yeah, wait, so we can do that?
yes.
Hooray! Okay, never mind. Yep. So that's one. Okay, yeah.
So, okay, I have a pretty cool story about UBE3A. But guys, this will be the paper I'll post it for. Actually, I think it's already in the folder. So if you go to your Journal Club folder on Google Drive, you can already take a look at it. I'll provide prints for the freshmen. Oh, wow. Yeah, so autism UBE3A, there's a big circadian connection here. Some of this was done, I think, in my lab again. But anyway, any questions, comments, concerns before I let you go? We held you a little too long.
I was gonna say, Char, if any of the freshmen or like underclassmen like really jive with any of the figures, like let me know and I can like designate that for them so I don't have to like cold call and be
Okay.
like, anyone want to do figure 6? And then it's like crickets.
Okay.
And also Maggie has a question. My queen. Okay.
Yeah. Meg.
Sorry, so I just had like a general question about the paper we just did. If if the cell has like a different like shape or size to it, does it affect like the virus, like how quickly it spreads or anything like that?
So if you look back at figure 6, kind of let me just put up real quick. You can see the different, it's like the high input, low. Yeah, there's the different ratios of output to input. So you see where there's more filamentous viroids, it's the lower output to input. And then on the right where it's those more spherical cells, it's or viroids, I keep forgetting to say, is higher output to input. So the spherical viroids again are better at spreading the disease and continuing to infect, and the filamentous viroids are better at, as Lila said before, defending against I guess problematic antibodies that's going to come across.
Okay, thank you.
Does that help? All right. Yep.
So, anything else? Was that torture or was it interesting?
Yeah. Sorry, I know that was long, guys.
I will find out from the freshmen tomorrow what they actually felt about it. I'm sure somebody will say something.
School
So-
tomorrow must be
I was
tough.
like,
Yeah. Imagine having school tomorrow.
Yeah,
Oh,
exactly.
yeah.
Have school.
Imagine having school.
amazing. That's crazy. There's a professional development day. That's why. Also, I just want to say my paper is so much cooler than his. So, like, if you didn't like it, like, that's too bad
Yeah,
for
it already
Colin, I
looks
guess. But like,
20
Professional
Yeah.
sleep day for me.
dude, they gave mice autism. You have to read it. I haven't even read it yet. I know
Alright.
that. You have to lock in. Okay, sorry. Anyway.
All right.
Mice. How did
Yo,
they
can
know
we
it has
leave?
autism? Yes. All right. Well, goodbye, everyone. Sorry,
Char.
Kate, to
Wow.
torture you. Goodbye.
You really didn't want to listen to me ramble, Lila.
What did she say?
She was like
She
begging
didn't
to
want to
leave.
hear you ramble.
No, that was it. That was all I had to say. Who was that? Who said
Literally
that?
scared
Yeah.
her